Hello everyone, I am Dr. J. Arunachalam from Sastra University, and I wish you all a very good morning, and I welcome you for this third day. I welcome you for this third day of the workshop, and I'm very happy to introduce today's speaker, Dr. D. Bharanidharan. Dr. D. Bharanidharan did his postgraduate in medical biochemistry at the campus of IBMS, University of Madras. Subsequently, he did his PhD in the Department of Crystallography and Biophysics, where he analyzed various aspects of computational genomics as well as DNA crystallography. You know, it was the first, it was the time when the first human genome came, all right? So he was one of the few people who started analyzing human genome at the very first draft when it came, all right? And later on, he moved to University of Alabama at Birmingham to work on cell surface proteins of various bacteria for his postdoc. And his second postdoc was in Georgia Tech, where he worked on cryo-electron microscopy and various computational aspects of cryo-electron microscopy. Right now, he is in Aravind Medical Research Foundation at Madurai, where he is working on NGS aspects of various ocular diseases. And I am very happy to invite Dr. Bharanidharan, and I am sure it will be a very wonderful lecture by Dr. Bharanidharan. Dr. Bharanidharan, you can take over from here. Thank you very
much. Thank you. Can you hear me clearly? Hello? Hello? Can you hear me? Hello? Dr. Arunachalam? Hello?
Yes, Bharanidharan, you can go ahead.
Yeah, I just want to check whether everyone
yes,
can.
Yes, we can hear you. You can go ahead.
Okay. Thank you so much for the nice introduction. Also, I would like to thank the organizer, Dr. Subramaniam, for inviting me for this wonderful workshop. Let me share my. So can you see my presentations? No. Yeah, so this, I guess everyone is seeing. The title of my today talk is Next Generation Sequencing Based Methods for Unraveling Genomic Variations. So as Dr. Arunachalam pointed out, myself, Bharanidharan from Aravind Medical Research Foundation. So before move on, I would like to take a few minutes showing this slide, what we have been doing at Aravind Medical Research Foundation. So our interest basically focus on identifying molecular signatures that can be used as a diagnostic and prognostic marker, as well as use the molecular signature to understand the pathogenesis of eye disease. Particularly, we are focusing on microbial keratitis, ocular cancers, TB, glaucoma, and other retinal disorders. Here we use most powerful tools. One is NGS omics, as we are going to see in this presentation as well. Here we use all kinds of methods like exome, genome, RNA-seq, small RNA, shotgun, whole genome. Also, we integrate, if necessary, with other omics methods. On the other hand, we use, we are very much interested on small non-coding RNA. Particularly, we are focusing microRNA, and we are extending to piRNA right now. Okay, coming back to the talk. So what I'll do, I will start with the introduction. What are the different types of genomic variations and NGS methods, past and present, and how this sequencing can be analyzed. And later, coming to the important step in NGS data analysis, variant prioritization. And after that, we'll move on to the examples on how we have been applying these methods to identify genomic variations in eye disease. And these are, and it's variant prioritization method. It's varies based on the disease. For example, cancer, as you can see, we are going to see retinoblastoma, one of the ocular cancer. If we are going to see Mendelian eye disease, how this variant prioritization can be done. If it's a complex disease, how this can be done. Okay, let's start with brief introduction. Okay, so everyone can follow later on. So this story you all know, right? This cartoon depicts that four blind men when they touches elephants different parts, and they think that differently. As you can see, one touches the trunk, it says it's a big snake. One touches the tail, he thought it's a furry mouse. What we are eventually trying to say in this picture is, when you have to get a complete picture, you should have all the information together. Like when four blind people, if put it together, then only they could identify this is an elephant, not different whatever they thought. So this is called omics. So we are not going to analyze or working on a single gene or single protein. Now it's like at the system level as a whole. So as I mentioned, omics, you can do at a different level in the central dogma. So if it's at the DNA level, it can be like the genomics, epigenomics. At the RNA level, it could be a transcriptomics, protein level proteomics, and the end product metabolomics. So we need to now understand all these systems so that one can understand the phenome, that is the pathogenesis of any disease. So the protein and metabolites can be studied using mass spectrometry or NMR. So in this presentation, we are not going to proteomics or metabolomics. So coming to the DNA and the RNA, so NGS can be used to study genomics, epigenomics, and transcriptomics. So in this presentation, our focus is only on genomics. So where we'll be studying all kinds of genomic variations using NGS methods. So I think it's clear, right? So you all know all this terminology now. So coming to the genomic variations, we simply call it variants. Let's start with the simple one, single nucleotide variant. As you can see, let's assume that it's a piece of polygon from genome. Say it's a reference, and then if there is a single nucleotide variation, we call it single nucleotide variant. And at the next level, it could be like more than one nucleotide base change. More than two or three may be inserted, or it may be deleted. It's called indel. So let's say more than that. Let's say more than 1 kb base pair is deleted, then we'll call it deletion. And if it's inserted, insertion, if it's inverted, inversion, and if it's duplicated, duplication. So all these four kinds coming under the structural variations. If it's less than 1 kb, then we call it cup number variations. So how all this can be detected? So that is the first question that we should answer, right? So Sanger sequencing is gold standard method people have been using for decades, and it can determine the single nucleotide variation at its exact position. Very robust. And later on, there is if you want to study all samples of genomes, genome-wide association studies can be used, but the limitations here is it can be used to detect only known single nucleotide variations, and mostly it's useful when we have case control studies. Also, it can be used to identify indels as well as CNVs, the copy number variations. So how about structural variations in the human genome? So there were several methods. Still, they have been using several methods. As you can see, I've put many methods like genome-wide scans, karyotyping, clone-based array, oligonucleotide-based array, SNP array, sequence assembly comparison, clone paired end sequencing. See, so each method has its own limitations, has its own advantages to identify or detect structural variations in the human genome. So all you have been seeing is different methods for single nucleotide variations and copy number and the structural variations. So whether we have any single approach that can be used to detect all kinds of structural variations in the genome. So that is where the next generation sequencing methods came in. So here we are going to focus on only genomics, and the genomics we have different NGS-based methods, whole exome sequencing, whole genome sequencing, yeah, and targeted sequencing. All methods can be applied to identify all kinds of structural variations like SNV, index, copy number, other variations. Eventually, it can also be translated into the clinical settings to better understand pathology, and also it will help to increase the diagnostic rate by identifying new disease genes, and also it will reduce the diagnostic time because it's a massively parallel sequencing, and obviously it will be helpful for personalized medicine. So as you can see on the title, I've put second generation sequencing for an extended sequencing. In a couple of minutes, I will tell you why it is. So coming to different NGS methods for genomic variation detection. As mentioned, whole genome sequencing, where we are going to analyze all, whole genome, all coding, non-coding, and mitochondrial DNA. So this method can be used to detect all kinds of variants, as I mentioned, single nucleotide variation, indels, SV, CNVs, everything. So, but it's quite expensive still now, and also the data analysis is still hectic, and also need more. It's more time consuming. So people, that's why I use whole exome sequencing, that where we are going to focus only on protein coding regions and not internal, because more than 85% of the disease causing these variants occur at the protein coding level. So that it's fast and cost effective sequencing method. And it could give more data, and also the data analysis has been widely, set for easy analysis. And there are only issue right now in whole exome sequencing is variant prioritization. That's what we are going to do, or we are going to see in this presentation, how this can be analyzed. And if the scientist or person who want to study only few genes, for example, in breast cancer, if you want to study only BRCA1 or BRCA2 genes instead of whole exome sequence, since we know that is the gene is involved in the disease, then they can focus or target only that exon region. That's called targeted sequencing, where you can increase the depth, and also it will be very useful for diagnostics. Okay, so how these methods, as mentioned, WS, WGS, can be done with the different technologies. Right now in the market, there are different platforms available. As you can see, Illumina, Applied Biosystems, and Torrent, Nanopore, Pacific Biosciences, Roche, and the new one, the MJ from PGI. Now Roche is absolute, and Illumina is dominating this NGS world because they hold more than 80% of the market share. So why is it so? And I will tell you in a couple of minutes. So this slide is important to see how these NGS methods has evolved, and as you can see on the left, I mean, y-axis is gigabases per run, and x-axis read length and log scale. So Sanger is here, and the read length is around 1000 base pair. As since it's the first gold standard method, we call it first generation sequencing. And the massively parallel sequencing has evolved over the period, and from the very short base pair length. After that, it's keep on evolving. So this next generation sequencing method, we call it second generation because we have third generation sequencing methods, MiniION and PacBio. So why we named it like second and third is because the based on this methodology they follow. Here in the all NGS methods, second generation sequencing, we have the step called PCR amplification. That is not required in third generation sequencing. That is a major difference between these two. Apart from this, each platform has their own methodology for sequencing. I will show you which one we mostly use around the world. So in third generation sequencing, Illumina, as I said, it's dominating the market. And recently from PGI, when China has developed a new method called the DNA nanoball sequencing. From 2016, it's evolving, and I put a box in the second generation because it doesn't require PCR-based amplification. Instead, they use like rolling circular amplification. So that is the main advantage of this one. So we don't know where to put, so I put it in second generation sequencing. The sequencing is more or less like Illumina sequencing, but there are differences. I will show you what it is. So I think now it's clear how different methodologies has evolved. Let's talk about the how the sequencing can be given done at the companies or wherever the lab has NGS machines. So this part is most important to understand everybody, because when you do data analysis, you should know all kinds of steps. So based on that, your data analysis also will vary. And if you are stuck with, or if you have poor data, then you still can dig out if you know what exactly been happening at the lab. So I have put two rows here. Actually, basically, the top row is the methodology followed by Illumina company, and the bottom row is MGI company. So these two, based on my experience, also if you go to the literature, you will see more than 80% of the publication from Illumina. Only rest of them from other platforms. Why I put only these two for because the application which we are going to see in this talk is genomic variation identification. If you go for third generation sequencing, you will end up with many errors. Though they are still this platform is evolving, the error rate is still high compared to Illumina and MGI nanoball sequencing. So for any genomic variations, if you want to detect, this is a preferred method right now. So that's how I put only these two. So let's see how this is being done. So in both cases, DNA, obviously, and in both cases, or any NGS method, the first step would be to fragment the DNA into pieces, because NGS cannot go for, and as you see, the read length is less than 1000 always. Only in the third generation sequencing, you can go, more than 10,000 base. So coming to the Illumina, so once it's fragmented, and then the next step it would be the DNA extraction and library preparation, where a specific adapter is attached to both ends, and that is used to attach on the flow cell. That's a chip actually in the machine. We call it flow cell. There are millions of packets, and then that they have already attached to a probe which is a complementary to that adapter, which we actually ligated to your template, DNA templates. So once it's attached onto the flow cell, it's the next step would be the clonal amplification. That's, as I mentioned, PCR amplification will be done. It's called bridge PCR, where all the each fragment will be, as you can see, like as you can see this amplified each fragment by the bridge PCR amplification method, and then after that each fragment you can see like a cluster of fragments. So then the sequencing will be done by sequencing by synthesis method. That's each nucleotide attached with the reversible guide terminator will be used to synthesize each fragment. Since it's a cluster fragment, you will get strong intensity of each color that is attached to each base, and then machine will treat the emitted color, fluorescent color, and then record based on the intensity. It will also give you the probability score called PRED score that you're going to see, which is most important first step for any NGS data analysis. So finally, we will be getting sequence data file called FASTQ, and I'll show you what it is in a minute. So this is the Illumina platform, and the recent one that they have evolved, from 2016. Now they came up with a very optimized method called RCA, rolling circular amplification. So then they make DNA nanoballs. So what it is as you can see, this first step as Illumina is fragmentation and then adapter ligation. And they also add a small piece of oligonucleotide, which will help and to make a circular uh, oligonucleotide. As you can see, each fragmented will be circularized using ligase, and then after that they use the special enzyme by the viral polymerase that can be used to amplify this each circle, and then it's continuously amplifying like circles, as you can see. That's why it's called rolling circular amplification. So after 500 cycles, let's say 200 to 500 cycles, it would form like a ball. So we call it nanoballs. Then that nanoballs will be loaded onto the flow cell, as I mentioned in the Illumina. And the flow cell, it's a patterned array. So the chemicals that coded on the patterned array, it's oppositely charged to the DNA nanoball. So each nanoballs can sit on the patterned array. And once it's settled down, and then after that, the sequencing will be done. And they came up with a new technology named School MPS, this flow-based sequencing, where actually same as each nucleotide will be attached to the each strand, and synthesis will happen. And but instead of reversible dye terminator, here each base is attached to the antibody, and it has its own dye. So that will be emitted and detected as Illumina, and finally we will get the raw data. So this is the two methods which is most commonly used, or you can use for any genomic variations identification. So once we have the data, that next step, as you know, that is data analysis. That is the focus of this talk. So if it's genome, it's a massive amount of data. You will see in each single text file in GB. So it's difficult to use a small machine. So you need a high performance computing, a big server to analyze the data because of several issues. So I don't want to go in detail because this can be, overcome by now various open source tools, various pipelines is available. Even we have our own in-house pipelines to analyze both the genome and exome sequencing data. So let's say what's the NGS data analysis workflow, for a generalized workflow for genome exome sequencing data. So as I mentioned, it's start with the FASTQ. That's a file that is from the machine. We'll be getting it. once we get the FASTQ, the first step for any engine is data analysis quality check. As mentioned, the probability score that's coming along with the sequencing reads its PRED score. I'll show you in subsequent slides what it is. That has to be checked. And then if it's poor, you can remove it, you can filter it. That is called pre-processing, and also primary analysis for any of these data analysis. For genome and exome sequencing data, and it follows the secondary analysis with alignment. We call it reference-based methods. So as you can see in this figure, so assuming that blue line is a reference genome, in our case it's human genome, obviously. And then we'll use the reads, whatever is coming out from the machine, a small fragment, right? But it's millions of reads. Each reads, as it's showed, PCR amplified and sequenced multiple times. So that has to be aligned against the human genome. So for that, we have several algorithms, like I mentioned, one or two here, BWA, that is borrower-based method, and no align. Another aligner can be used to align these small fragments that reads against the reference genome. This is called reference-based mapping. Without reference, you can build consensus sequences, that is called de novo assembly, but that is not the deal here. So whatever we are using in genomic variations identification is only reference-based map. So once it's mapped, all your reads against the reference genome, then the final step would be the identify the variant, and variant prioritizing that, which find pathogenic variants, which is the disease causing variants. Why is it so difficult? Because yes, because whether it's whole exome sequencing or genome sequencing, you will be getting like thousands, like more, at least if it's exome, 20 to 30,000 of variants you will end up with. And if it's genome, again, you will end up, even I'm talking about only the single nucleotide variation, you will end up with more than 60. So if that is the case, so we need to filter, right? Filter 60,000, and you have to nail down to particular one or two variants. If it's Mendelian disease, if it's a complex, more than few. So that is a big task. So that is what we are going to see in this presentation, how this can be done. Okay, let's go into the detail so that you will understand when I go, whenever we go along the presentation. So this is what I've been telling from the beginning, FASTQ format. Basically, it has first line, it's all text file only. The first line is machine identifier and all information about the sequencing machine, and when it's done, everything, flow cell information. The second line, apparently this is the sequence. It's small oligonucleotide. If it's NG sequencing, it's not more than 300 base per length so far. And the third line is some ASCII symbols, because that's how the machine used to give. That's called PRED score. Basically, what the PRED score means is just its probability score. As you can see, it's a log value of probability score that comes out from the machine, and it's converted into PRED score with ASCII values. As you can see, this ASCII character, if you see in the read, it tells that its error probability is 1 to 0.13. So what we require for any diagnostic purpose, or if you want to study, we should have at least above Q30. That's why Illumina gives you mostly 90 to, more than 90% of the reads are above Q30. That is PRED score, Q30, and the error probability is 0.001. That is less than that percent of error rate. So you can trust whatever that coming out after alignment, the variant calling, variant list is the true positives, not the true, false. And since different platforms and different methods giving output, there are also difference in their file format. Basically, in the ASCII, character only. As you can see, 0 to 40 is the Sanger method. We call it pride plus 33. Basically, the number of ASCII character is only 40, and if it's Illumina, there are different versions. So when you do data analysis, you should be careful about these parameters. Now, I think I have not updated, I guess recently, there are 1.9 also for Illumina. So this base, if it's different one, for example, if you are using the world aligner, if it's not updated, you are using different FASTQ format, then you can convert to whichever format you want for the data. So these are the very preliminary step, or a very important step to at the level of quality check. And some consideration when you go for, as already mentioned, if it's exome sequencing, you can use a small workstation. If it's whole, genome sequencing, you need a big machines. If you want to do it yourself, don't depend on Windows platforms. You have to depend on Linux platforms. That is very obvious because most of the open source tools have been written in Linux. Or you can go with commercial software. Any commercial software can run on both Windows and Linux. So there are some consideration when you do data analysis. And also you should know when we do data analysis, there are also some hiccups, right? Always, because each platform has their own limitations. As mentioned in NGS methods, they can go above 300 base per length. So at the end, more length, and you can expect more errors in the NGS-based methods. And if it's homopolymers or repeats, there you could always expect there could be errors. So when you do data analysis, this all you should remember, and that can be sorted out, or you can with the data analysis you can eliminate all these limitations. That is pretty straightforward. So I'm showing again the example how this pre-processing can be done. If it's, as you can see, let's say this is a 100 base per length data which we got from Illumina, and as you could see, the y-axis is just the Phred score, as you showed. The Phred score value, the green part is about 20, and the yellow part is about 20. Green part is about 30. So all I want for a diagnostic purpose about 30. So I don't want whichever that is less than 30 per score. So there are many tools available. You can use one of the tools to remove it. I will show what we have been doing. So after that, as you can see, we removed all the reads which has low quality. So there are some problems if you go for small RNA sequencing or RNA sequencing, because the kit they use is as which could form an adapter dimers. But whole exome sequencing, you won't see such big. Most of the reads, like as you can see, this part is the adapter contamination area. If it's a whole genome sequencing, you wouldn't see that much. You will see always like a small percentage. So if you see such data, then you can't use it this part, less than 10% of the data. So you should throw out. That lakhs of rupees will be wasted. So you should see that also, adapter contamination, any repeats, oligo, or even your primer, hope, whatever you use for ligation. That can be removed easily if it's less than 10%. As you can see another one. This one I already spoke about, what is reference-based mapping. As after mapping, this is how you're going to see. You can manually check also like that. And the point here you have to note down is the human reference genome. As we talked about earlier, human genome project. After that, there are many reference genomes. Currently, there are, 2 reference genomes available. Recent version is HG38, GRCS38 from different databases. So this why I'm pointing out here. Some people, some company use kits. I mean, probe capture if it's exome sequencing. That probe capture is designed maybe a previous genome like HG37. In that case, you should not use the current build. So you have to be careful when you use which human reference genome you are going to address. So for alignment, the next step, there are multiple tools as I mentioned. So there are many tools available as an open source. So it's very easy. You can identify which will work best for Illumina. And we have also benchmarked it, and we have also uh, published it which tool will provide most, I mean, true positives, I mean, compared to false, positive and negative. So I highlighted two that we have been using this based on our benchmarking results, NovaAlign and BWA. These are two alignments we've been using it. So these are all format you should understand when you do data analysis. SAM, sequence alignment mapping. Uh, this is how we used to get after alignment. Any tool you use, these are the format. And then this is a big file, so you don't carry for the next step. So you have to convert to a small size file that is called binary version of SAM. So tools are available that can be used to convert this format, and finally bed format may be needed, as you can see, for a probe capture method. Like as I mentioned, both targeted sequencing or exome sequencing, you need this file to where you are going to focus that identifying genetic variations at particular area. So coming to the variant identification, that is the third step. So it's based on your disease type, also where, I mean, what is your interest? For example, if you want to identify only germline variations, somatic variations, or only CNV or SP. So as I mentioned, if you working on it, lymphoma, cancer, any cancer like leukemia, lymphoma, you are not going to expect germline, you will expect only somatic. If it's BRCA1, if they wanted to detect early, then you are looking for germline variations. And some cancers where we know only copy number variation can be identified and can be used for a diagnostic purpose, then you can focus on only CN, so that you can change the NGS method as well. Instead of doing whole genome sequencing and spending more money and for both sequencing and data analysis. And also what we require is a sequence coverage. If it's a diagnostic purpose, you should have at least, I mean, more than 100x coverage. That is how many times each nucleotide is sequenced. And NGS tools, obviously, what kind of tool you're going to use. So for variant detection, there are again there are many tools available as open source. So this is the one very widely being used for a long time, that is called GATK. It is machine learning methods, obviously. And there are also many tools recently been developed. They perform even better than the GATK. So the one is by Google, that is deep variant, which also won the award that they can be taken up for the precision medicine, because the overall F score, that is what F score means, it's both sensitivity and specificity combined together. It's more than the GATK and other tools. So once you see such variant callers, you will get the final file called VCF file, variant calling file. And that file has several informations that is coming out from the each tool, as I mentioned, deep variant or GATK or Strega, that one developed recently. So as you can see, this format for each nucleotide, there are multiple parameters. You have to be very careful, and also which one you want to use it for filtering. That is, as I mentioned, in whether it's exome or genome, you will end up with more than 20,000 variants. All will be in this file. So you have to filter those variants. For example, common variants. If it's in, let's say, Indian population has more than 10,000 variants without affecting your functions, then we don't need those variations further. So we can filter out if it's a common variation based on the frequency. So such information can be done after annotation. But in this file, you will see all uh, that parameter that talks about the quality of the variation. So that can be used to filter it. For example, PRED score, as I mentioned, that will be there. If it's, as I mentioned, coverage, if it's less than 50x, if I don't want I want 100x, then you can set, you can write a simple script, or using Excel, you can filter all kinds of variations. As you can see, genotype quality, read length, read depth, sorry, haplotype, that is whether it's homozygous or heterozygous. So this information is very useful when you do data analysis. That is the first step to filter. I want only the variance, which is always true positive. Then you can apply all kinds of parameter to filter out. This is the one we commonly use. So each lab has their own recommendation for filtering. So for SNVs, we apply such parameters like mapping quality 40. That means how we are mapping. If it's pattern sequencing, that forward reverse sequencing Illumina, how it's mapped, and then there will be a score after the variant calling that can be used to filter. And QDS is a quality score and depth divided by depth. Similarly, you can use several parameters that comes out from the VCF. So if you Google it, you will identify what are the different parameters one can apply to filter out variations. These two I have put it here because this is a parameter we follow for single nucleotide variations as well as indels for both human genome and exome sequencing data. So the final step after initial filtering of the variations, functional annotations. So why we need it with this base VCF file, as you can see, it will give only matrices about the variations. What about the variations we detected from the human genome or exome sequence. All we want to know where actually they are located and what is this function, everything we have to know for further variant periodicity. Then only we can say whether that is really disease causing variant or not. So that is still long way to go. So let's see what is the functional annotations. So once we get the variants after variant calling pipeline, you have to annotate whether the variant, where it's actually located, it's promoter region or coding, or if it's intronic, if it's a spice junction. All we can annotate using several tools, annotation tools available. So there are again multiple tools for annotations. This is the ANOVAR is one annotation tools we have been using for a long time, and it's quite good also. So I would recommend you can use ANOVAR or any other tools. Since all the tools have multiple data, they presently they use multiple database to annotate your variant. It's not only as I mentioned earlier, but now they've included every month more and more databases to annotate your each variant. As you can see, it can be annotated whether it's in the microRNA, if it's in the binding site of if it's a methylation site, and also it will predict whether it's deleterious or neutral variant. And there are several databases now available, expression databases available. If the gene is highly expressed in particular disease, you will get it. So if it's in the cell line, it can also give whether it's detected in cell lines earlier. So now ANOVAR giving all kinds of information of particular variant. So what is the next step once you annotate it? Is it easy that you can filter out or to prioritize variants out of, let's say you started with 60 from exome sequencing, after filtering 20,000, after the annotations, you can see around 20,000, maybe hundreds. That won't help, right? So basically, we need to distill the exact disease-causing variant for a particular disease. There is another issue. You should always remember that any individual, if you do genome sequencing, you are, let's say, he's a healthy individual and no disease, no hereditary disease, and you will always end up with at least 50 to 100 disease causing. They are sitting, maybe later on you will develop a disease, or it may be a late onset disease causing variant, but we don't know. So that is the big question we have. So you will always see, you will always find out the variant, that disease causing variations after the end of the analysis. Then there another big question, which one actually, if it's a person having any disease, among the 50, which one is actually leads to a disease is a big question. So how we deal with, in case of eye disease, I will show with some of the examples that this can be applied to any disease, but it requires knowledge of a particular disease. So this is the final step, and it's the greatest bottleneck now to distill to narrow set of disease causing variants out of bunch of variants. So here we applied different theories, or I would say hypothesis based on your disease, what kind of disease you are going and to analyze that you are interested in. So I put categorized all disease under three categories. Mendelian, as you all know that, Mendelian disease, common disease, as you all know, diabetes, and other cardiovascular, there are many common diseases. And rare diseases comes under like very rare, for example, like retinoblastoma. Among the other cancer, it's 1 in 10, 15,000, so we call it rare disease. So here, what kind of methods we need and how many samples we need to distill down, to particular pathogenic variants. If it's like say Mendelian disease, as the plot depicts, that you don't need many samples to be sequenced. Let's say if it's a recessive disorder, if you do it father, mother, and the child, that is just 3 samples. Doing a WES is enough. You can nail down to disease causing variants. So I'll show in some of the examples. If it's a rare disease, we'll go between 10 and 100. If it's common disease like diabetes, so we should have 100, because we need case control studies, and we have to follow various statistical tests like burden test, so that we can identify the common variations that can be identified. So coming to the WES, if you want to use only WES, you don't want to use WGS. So what kind of hypothesis one can think of? So one is single gene with homozygous or heterozygous mutation that can be easily be identified if it follows a Mendelian pattern of disease. And also extremely rare variants only cause disease. That is a hypothesis that we always can think of if you want to use the WES for a disease, it should not be a complex disease. So there we can apply that extremely rare variants only cause disease. So large effect size and completely penetrate. That means if the variant is there, it should lead to disease. If it's a common disease, it need not to develop a disease. So it should, the penetrant means whenever the mutant is there, it always causes the disease. So that hypothesis, if it's there, you can just go for WES. And also it always affects the protein structure and function. And like one I said, it can change the spike site, and also one base pair deletion insertion in the coding region will lead to frame shift, and the entire sequence will be changed, protein structure and function will be altered. So how this all can be filtered for a particular disease? As I mentioned, different disease requires different kinds of filter. So this is the common workflow that we follow. Let's take a look. Now from the after the VCF and annotation, as you can see, I've put only 3 diseases, which I'll be talking about today with an example of eye disease. So the first one, cancers, complex disease, Mendelian disease. The MAF is the multiple allelic frequency that, as mentioned earlier, if it's a common variation among the population, for cancer less than 1% may occur. For example, breast cancer, if it's a BRCA1 mutation, you can expect less than 1% or more than 1% in the population. If it's a complex disease, this will be among the population that variation which we are interested in may be present in more in the disease, in the population. So it's difficult to identify, so that we have to increase the multiple allelic frequency. That is, they are more common in the population. Like diabetes, the variations may be there, and then they will develop a disease later on or may not. So that is the penetrance, low penetrance. In Mendelian disease, like a cancer, it's always be rare variant, as I showed earlier, and is as expected less than 1% in the population. Then it would be the functional variant filtration. As I said, stop loss, stop gain. That any variation that introduce any stop gain or stop loss. That means that if it's introduced stop codon, there won't be any translation. You all know, right? So if that is the case, then it's obviously we know that that's going to affect the protein structure and function. So that's first step to filter functional and based analysis. First to filter the variance based on its functional effect, like frame shift, stop gain, stop loss. If it's going to alter the splice site, that is also be counted as a functional variant. So then the other, that is most of the disease causing variants not comes under this category, which we can easily be filtered out. That is missense variant. That is single nucleotide variant which affects the coding sequence. Just that we call it missense variant. If it's altered, only one amino acid. So in that case, we use different methods, machine learning methods, because it's a very tough to, a difficult task to filter out whether it's really disease causing or not. So in that case, we apply several parameters, not only the variant information and the gene information. We also use several other prediction tools. If it's a cancer, we use cancer database to predict whether they are mostly presented in cancer patients. If it's a complex disease, we can use segregation analysis, case control statistics like burden test, other tests. If it's a Mendelian disease, we can use how this variant is inherited. And if it's trio or if it's autosomal dominant, we can also use the segregation pattern of the variation along with the phenotype. So all this I'm going to show you one by one with an example, so you can understand better. At the end, even if it's a difficult task, we still can use other databases. Basically, like if a gene is highly expressed in eye tissue, not in other tissue, then we can focus only that gene variations, not other gene variations. So such information can also be used to filter out and prioritize the variant. That's why I put last part as a knowledge based, and first part is heuristic filtering. In between the phenotype based predictions, I put in-house where we use machine learning methods. So this all about we are starting from the NGS methods, and then how this data analysis can be analyzed, and what are the different steps. So common for exome genome sequencing data, and at the end how variants is prioritized or filtered and prioritized based on the disease phenotype. So this is a general scenario. Whenever you want to apply for any disease, this is how you can follow. But when it comes to a particular disease, then you can, change a little bit basically at the variant periodization level. Other steps you can follow, as I mentioned. So in this presentation, I'm going to show some of the example with what we have published or what we have been working. So our focus is on eye disease, genetic eye disorders. This is simple picture depicts the anatomy of the eye schematics where you can see this eyeball, and this is the anterior part, and you will have a transparent layer for cornea, and behind that you have a lens, and after that it's a big chamber filled with fluid called vitreous humor, and the back of the eye we have that red color pigment loaded cells called retina, and then we have choroid, sclera, and several tissue, optic nerve, and so we don't want to go to the details. So, but what I wanted to focus here, we have usually call it this part is anterior segment, and behind that is posterior segment. Why I want to separate the eye into two is anterior segment has different type of genetic eye disorders, and posterior part again has. So I've put numerous genetic eye disorders, right? You cannot imagine that you don't see, right? Much might have seen some of them, like the one common, that is a cataract. I put here, it's also coming under genetic disorder, anterior segment, and have list of genetic disorder here and also here. So this all the genetic disorders we have been working on. So our genetician been working in this number of disorders for a long time, more than decades. So I'm going to show some of the examples that where actually we have applied NGS methods to filter out or prioritize the pathogenic. This is causing variants for a particular genetic eye disorders. So let's start with the cancer. So this is called retinoblastoma. This usually occurs less than 5 years of child. As you can see, this can be a both eye or one eye, and sometimes it's be a very pathetic. And if it comes to a late stage, all we want to bring the child very quickly so that it's 100% curable. So we always be just giving this. Just take a look at this picture, the white eye. So if you see, even if you take a picture, but now all mobile cameras don't pick up this white, right? Because the mobile cameras has automatic red eye reduction, but that's not we require. We need to see whether if it's a red eye, that is eye is normal. If you see white, then there will be a problem. I mean, could be a other eye disease. Most probably it is a retinoblastoma. So even if you find any pictures, any child with this, you should bring the child to the hospital as an emergency basis. Okay, so what we are going to do in this retinoblastoma, the genetician, that it could be a heritable disease, like a breast cancer, and most probably 80% of the mutation occur in this gene called retinoblastoma gene 1, and it could also be a non-heritable, so where it can occur as a sporadic variations. So on the other side, as I mentioned in the study design, germline mutation in this cancer, it can also be a somatic variations. This is the gene we expect always retinoblastoma gene. 85% of the mutation occur only in this gene. 15% we don't know. So that's where we usually apply NGS methods to wherever we see unknown cases after standard sequencing of RB1 gene. So for retinoblastoma, we follow this pipeline. So this generalized pipeline I have already showed you, but for retinoblastoma, we follow additional tools, as you can see. This aligner is same, as I mentioned earlier, BWA, and pre-processing, we use these two tools, FastQC, Cut Adapt, and here mostly we use Illumina, and recently we have started using MGI nanoball sequencing also. the data of nanoball and Illumina are giving no more than a PRED score Q30. So in this case, as mentioned, we wanted to identify germline as well as somatic as well as copy number variation. These three kinds of genomic variations we always expect in retinoblastoma case. So we can use exome sequencing or genome sequencing, since it's 95% of the genetic variations will occur in this type of variation. So we always prefer whole exome sequencing. So as mentioned earlier, it's based on your study design, what you are expecting. So in this case, we always use exome sequencing. And the first thing is to focus on RB1, where we always expect 80% of the mutations. And sometimes it could be a somatic, where we have to use different tools, so that to detect the somatic variations, we need both a blood sample, and the tumor sample. Then only we can identify somatic variations. So that based on your interest, its sample input and number of sequencing of sample also differ. Also here we want to study copy number variation. So we use different tools, variant calling, SNV, and indels. You can use the variant ZATK as mentioned earlier. With somatic mutate is another tool. If it's a copy number, we are using two tools. Since we in exome sequencing, we always expect more false positives. So to avoid false positive, we use multiple tools. After that, annotations. Since it's a cancer, we use cancer database. And then finally, sometimes if it's less number of reads, as mentioned, coverage should be high, since it's all going to be reported to the patient. So we have to be very clear in this case. So we always double check with the additional method like Sanger or endoscopy. Finally, that disease causing will be reported. So this is what I mentioned. When you go for a somatic variant, you should always have compare germline as well as tumor tissue. Germline means the blood, you can use it as to detect germline variation. When you compare, as you can see, same patient blood and tissue, in this case retinoblastoma tissue from the eye, and germline this blood. So once we do the sequencing, you can see that somatic variations can be identified like that. If you compare it, the person same blood and the tissue, you will see these kind of. So when you do manual examination, you will identify that there are some variations in the tumor that is called somatic variations, which is not present in the normal tissue. This is one example that based on the recent results with, I would say, maybe 20 exomes of retinoblastoma. As you can see, we could identify so many variants, all kinds, structural variations, including copy number variations, missense, that is, which alters amino acid, and altered splicing or create a cryptic splice site, frame shift indels. So if you put together, and then it actually we arrived at 97% sensitivity, also diagnostic rate at 85%. Still lacking 15%, right? So that we are working on it, that we could use a genome sequencing to identify. Okay, so that's one example for cancer. Right? So coming to the Mendelian disease. So you all know, right? Mendelian pattern, that's one gene, and that's now it's not always true, right? One gene, monogenic disease in Mendelian case. We always you can also expect it could be a more than one gene. It's called digenic disease, or more than two oligogenic disease, but all follows typical Mendelian patterns. So for Mendelian disease, the hypothesis which we follow, and we can expect rare variants can occur with highly penetrant. That is, whenever they are there, they can cause a disease. So if so, then we can use such model for to do WES for variant prioritization. So one is a linkage-based strategy. If its variant is running through family, you can follow and do sequencing WES of the family affected family members. And if it's a homozygosity based strategy, you have to do both father, mother, and the unaffected person. If it's a double pattern, also you can follow the same. Sometimes it could be a overlap based strategy, that is two different nuclear family you have to do both. Let's say in one family it's homozygous, one is heterozygous, so it can also be happened. So you have to do trials again and de novo based strategy. So both father mother didn't carry, but only the uh, daughter or son developed. Then we have to follow de novo based strategy. So candidate based, we don't have money, we can't do all trials, just do the one person with the disease and try to identify. So these are the various strategy one can follow for mental health disease. And also variant filtering. This is also our this one I already mentioned it in the previous slide. How variant filtering can be this same we follow for all the ITC. But when it comes to the variant prioritization, we follow differently. Right? As for a cancer, I showed you earlier, but for a exome data analysis, especially for Mendelian disease, we again use particularly the inheritance pattern and the segregation. That is not, we are going to use it for cancer. Okay, so let's start with one example, monogenic Mendelian disease. It's called liver congenital amaurosis. This is one of the retinal dystrophies. So we have been seeing in our hospital more than 200 retinal dystrophy disease. This is one kind. You can see it's a progressive nature, LCA. If you don't treat at the early, so you will lose vision, which is irreversible blindness. So this picture shows how this affects. These are like clinical picture how this affects, how they diagnose clinically. And this is the pedigree. That is a family chart where we usually based on the disease pattern we can draw it. So in this case, this is the person came to the hospital with a disease, and we wanted to see whether it's this any mutation running through the family. Since LCA is rare, I mean, autosomal recessive inherited disease. Recessive means the person must have a homozygous variant or compound heterozygous, and one variant may be from father, one may be a mother. So, as I mentioned earlier, the strategy can be followed as a trio sequencing, right? So we have done that. In this case, exome sequencing of all three, father, mother, and the proband. After sequencing, as I mentioned, the raw data gave us 63,000 variants. All I want is only one, since it's a monogenic disease, one variant only have one, that should be a homozygous, right? Or a compound heterozygous. So then eliminate, find out exonic, eliminate all the common variations and identify whether they are deleterious variation. And finally, we didn't get any functional, which can immediately we can detect like stop codon, gain, splice. We got in this case 40 deleterious single nucleotide, missense variation. So then we followed recessive mode of inheritance. Since this is autosomal recessive, so we followed the mode of inheritance, and then we got 8 actually. That is 8 disease causing variants. All are related to eye disease. So, but what I want is only one gene, that is monogenic. Since it's monogenic Mendelian disease, I want only one variant in this case. Hello. Sorry. I have some issue. Sorry. Hold on a second. Hello.
Yes, we can hear you.
Yeah. Can you see my slides?
Yeah, I can see the heuristic filtering approach, monogenic.
Yeah. So I have some issue with my monitor. Let me connect. Sorry. Okay. So anyway, I can present without seeing my monitor. So this we are talking about LCA, monogenic disease. So here, um, so finally we use machine learning methods to prioritize among these four, right? Four variant which we got, I mean, eight recessive genes, homozygous variant, and then based on the score, phenotype score, we finally prioritized one genetic variant that could cause the LCA. And then we validated again with the Sanger sequencing. That is one we could see it here. And move on to the next. Uh, I cannot go to the next slide. Is next slide is moving yet? I couldn't see.
No, you're still seeing the same slide.
Same slide. Issue with my it's working. Can you present the slide so I can share it?
Yeah.
Yeah, let me see.
Send it by email.
Yeah, I have it. Yeah, send
Send it
to the conference email ID.
Okay, it's 2 minutes. Yes.
Is Zoom link we have to restart after 45 minutes? Is there a restriction?
Sorry, what, Bharani?
Usually Zoom has only some restriction. We have to restart after 45 minutes.
No. This is a professional version, so there
Professional.
should not be any restrictions. Okay, so I will.
Yeah. Still my computer couldn't.
Okay, but you can proceed.
Yeah, okay. Yeah, next slide. Yeah, thank you. Okay, sorry for that delay. But anyway, we can finish it in time. So I have a few more slides on. So far I've showed you that monogenic Mendelian disease, how variant prioritization can be done. So coming to the digenic disease, yeah, this is again a retinal dystrophy. It's called Usher syndrome. The symptoms are more or less same, but it affects a different area of retina. So in this case, as showed earlier, the general workflow follows from the raw variants and exonic, common variation filtration, and then deleterious missense variation prediction. And we end up with again 8, and also all follows the recessive mode. And here we couldn't go further, that even if you use the machine learning methods of ours, it showed two or three genes, and which is not more relevant to the disease. So when we check back the disease nature, this show many literature show that it could be a digenic disease. Then we applied database called DIDA, which has a digenic path. So based on that filter, when we applied, then we could identify the digenic path. That is 2 genes that is involved in this disease. So all I want to say is here you have to follow that the knowledge also. If disease is monogenic, if it's digenic, and you should know before variant prioritization. Okay, next slide, please. Can you go to the next slide?
Already, I can see the next slide.
I'm seeing from my mobile. Okay, so so far we have seen both monogenic, digenic. Is this approach is realistic to detect disease-related variants so easily? Because in both cases we applied a simple strategy. Right? What if more number of deleterious variants detected that seem to have no pathologic effect, and we are not targeting rare but common variants. As I showed, if it's more than 1% of my multiple allelic frequency, what happens if it's more variants are segregating in the family? As I showed, if even those trios, more variants in segregating, then we can't filter easily. And what happens, many genes or combination of them are involved, then we have to apply different strategy. Can you go to the next slide? Next slide. Getting slow internet. Sorry.
Banidan, can you stop screen sharing? Because
Sorry?
Can you stop your screen sharing? Yes.
I couldn't because the monitor got stuck.
Oh, okay. So let me try.
Wait.
Stop sharing. Because now both of us are sharing, so it's
maybe I can wait.
Stop this.
You have a
More button.
but you just did, no?
just.
I guess only a few more slides.
Okay, I think you can proceed.
You can?
Yeah, let's proceed. I think we can.
Okay, can you change the slide?
Yeah, I can, but I can see it in the. Well, his computer is froze.
Okay, so I can talk. Maybe you can share the slides.
Yeah.
Candidates. So the next step, when if we fails all these hypotheses for WAS, as I mentioned for monogenic and digenic disease, if it's a complex disease. In this case, I'm going to show one complex disease called glaucoma, which is a because of high pressure, intraocular pressure, which develops damage. Which leads to the damage to the back of the eye and leads to the disease called glaucoma. So this is a complex disease like diabetes, so where we can't expect one or two genes, and also the hypothesis, whatever I mentioned for WS, all will fail. So in this case, we have to go for more exome sequencing. In this case, we have done more than 20 exome sequencing. And then after that, follow the same filtering approach. But when it comes to the prioritization, it is not the same as for monogenic or digenic for cancer. So in this case, it's a complex disease. We can expect it could be a common variation. So we increase the allelic frequency, that we can expect the variation could be more common in the population compared to the control. So when we have case control population, so we apply. We don't filter all the common variation. We filter less than 5%. We usually start with, and then see whether we can find any disease causing variant. And the next thing is to apply all mode of inheritance. If it's since glaucoma can be autosomal dominant or a recessive disorder, so we apply both strategies whether we can filter out. In this disease, even after doing all kinds of filtering, prioritization, we didn't find. And then we have applied our machine learning methods to prioritize RDC gene-based model. Disease genes only. In that case, still we got more than 20 genes. So among the 20, we don't know exactly whether all 20 is involved in this glaucoma disease or not. So then we have applied, as mentioned in the workflow, that overview of workflow, where we can apply other strategies like protein-protein interactions, and whether if they are under particular pathway or a particular biological process. So we have applied all other strategies, and finally we have found most of the genes which we found from the exome sequencing all comes under three pathway, which we know that these three pathways mostly been involved in the glaucoma pathogens. Then we focused on only three pathways, and then finally we end up with 10 or 15 genes. And again, we have to prioritize, right? We can't say all 10 or 15 is involved in disease pathogenesis. So we applied multiple information like GO, terminology, PPA interactions, and also the pathway. So finally, based on our machine learning, and then we have filtered out six genes, and then when we check back to the pedigree, and then all six actually sit properly with the segregation in the pedigree, and then finally we have arrived at these six genes may be involved in this family, not in all the family members, which we don't know. So we have to do more, maybe a genome sequencing may help, but based on the exome sequencing, at least we could derive at one a set of family, subfamily in the big family, following this, pattern of six genes may be involved in the pathogens of glaucoma. So that's all. And final slide is, I don't know whether you could share it, is the acknowledgement slide. So we've been getting funding from mostly all the agencies of the Government of India, as well as the private from Aravind Foundation, Sun Pharma, Cognizant, to do all our eye disease projects. And here I should thank our clinicians specifically, because they are the one actually bringing these problems, and also they are the one will provide us the samples, and also other investigators as well. Thank you. So sorry for that last few minutes we couldn't share.
Thank you, Banjan, for this excellent talk. We do have 3 questions that participants asked.
Okay.
For bacterial whole genome sequencing, which method, according to you, would be suitable, cost-effective, and would give best coverage?
It's bacterial genome sequencing. We have not talked about in this presentation. Anyway, we have been doing it. For bacterial genome sequencing, again, it's based on your requirement. If you want to identify a pathogen, if it's an endemic or pandemic situation, if you want to identify a particular strain, you can go with mini ion sequencing or PacBio. But you can't expect all the variations in detail. You can wait. That is again, but it's not cost effective. I wouldn't say. I would say still Illumina works best for bacterial genome sequencing and MGA as well. So compared to price, Illumina and MGA giving less price compared to Illumina. But MGA has recently started their market in India. They have not exported it in every part of the India. So you can go with the Illumina sequencing. If you want to identify only the bacterial pathogen, and then you can go with the mini ion sequencing. Any other question?
There's 2 more questions. So is it reliable or suggestible to use the genome assembly tool or of the Patrick database?
Sorry, last.
Sorry. Is it reliable or suggestible to use the genomic assembly tool of Patrick database?
For a genome assembly, there are still reliable, but there are other best tools also available. You can use it. The database as an assembly tool or as I think it has already sequenced assembly also, I guess.
Okay.
For a bacterial genome assembly, you can go with commercial tool like CLC Bio. If it's freeware, there are multiple tools or exist for a particular sequencing method.
Okay. The third question is for reference-based alignment, is it important for the reference genome to be complete? For example, the reference genome, the nearest phylogenetic neighbor of our bacteria of interest is present, but it is not a complete genome. Then what should be done?
Now, when if you want to do new strain for a bacterial sequencing, you don't have to do reference-based. You have to go with a de novo assembly and make a consensus sequences. If it's Illumina, will be getting only configs, the complete genome. If you want a complete genome sequencing of your bacteria, I would recommend do both hybrid sequencing, Illumina as well as mini-ion or PacBio. So their error rate will be high. That can be patched up, polished by Illumina reads, so that you will get complete genome sequencing with accuracy, but not the reference-based sequencing, only de novo assembly.
Okay, thank you. So if anybody else has any other questions, so you can either type it in the QA box or you can unmute yourself and ask the question directly. Okay, I guess there's no more questions. Then I will take this opportunity to thank Dr. Bharanidharan for his excellent talk, introductory talk for NGS methods and his own work. And okay, thank you very much. We're sorry about the technical glitch towards the end. And once again, we will send you the PowerPoint slides so you can use it. Okay. Thank you.
Just 3 more slides on this. So please, thank you so much. Thank you for your patience.
Okay, so that concludes our first session. The second session would be a hands-on session that will start at 11 o'clock. So please take a break and rejoin the meeting at 11 o'clock. Thank you very much. Thank you, Bharanidharan.
Thank you.